Hello everyone, welcome to C Academy YouTube channel. My name is Veleleni Nkosi. In today's lesson we discuss the examination guideline regarding meiosis. The examination guideline document tells us the things that the examiner wants us to know before we sit for an examination. If we want to get good marks in final examination, we need to understand the examination guideline. Then in this video, I will break it down to you in details. Without further ado, let's get to it. So this document that you see on your screen is an examination guideline. It's from the Department of Basic Education since it was done in 2021. So this one is regarding meiosis. Meiosis falls under paper 2, which is 21 marks in term 1. So these are the content that the examiner wants us to understand. So I will break it down to you in full details, and then I've already done some videos about this content, so I will put a link in every section. So let's start. The introduction. So these are the things that the examiner wants us to know. Number 1, we need to do the revision of the structure of the cell with an emphasis on the parts of the nucleus, the centrosome, and the cytoplasm. So this is the number 1 thing that the examiner wants us to know. So to explain it to you, this is an animal cell. You remember we did animal cell in grade 10. And then, so in this chapter, we will focus on the part of the nucleus, which is nuclear membrane, and the nucleolus, chromosomes. These are the part of the nucleus. And another thing is the cytoplasm. Cytoplasm is the space inside the cell. And then centrosome. The centrosome, these are this part. We've got the centrosome, which is 2 centrioles, is this part, and then on the other side we have another one. These are the things that we will be looking at in this chapter. So we must be familiar with this part of the cells. So another thing that the examiner wants us to know, we must know the structure of a chromosome. A chromosome consists of DNA, which make up genes and proteins. So this is the simple structure of a chromosome. This is a chromosome. If you can see here, it's wrapped. These are the things that are wrapped, and then we have DNA. We learned about the DNA, and then this DNA is wrapped around a protein. The name of this protein is an histone. So as a grade 12 learner, you don't have to worry about this name of the protein. You just have to know that the DNA has wrapped around a protein. So the name of the protein is not necessarily. So when the DNA wrapped around the protein, this section, we call it chromatin network. So this chromatin network become more wrapped and become more condensed, then it makes a chromosome. So a chromosome is this structure. This structure is double thread chromosome when it's double like that, or replicated chromosome. And then if it's single, maybe like this. Sometimes they will give you, like, you find that it's single like this, and then this is single thread chromosome. So it's not yet went under a replication, DNA replication. So you must be familiar with this structure. You must know that a chromatin network, it's a DNA which is wrapped around proteins. Okay, and then another thing that the examiner wants us to know, we must know the number of chromosomes in a cell is a characteristics of an organism. For example, humans have 46 chromosomes. So here we have a cell. This is a cell. Inside there are chromosomes. As human, we have a pair of 23 chromosomes. 23 from paternal and then 23 from maternal. So these are pairs of chromosome, like one is from my father and one is from my mother. So we must know, as human we have 23 chromosome. These are the number of chromosome which are in pairs. So this thing, we call it karyotype or human karyotype. So it shows us the number of chromosome that are in an organism. So as human, this is what makes us being a human. So another thing that the examiner wants us to know is chromosome which are single thread become double as a result of DNA replication. Double is a two chromatins joined by a centromere. So here this statement tells us that this is a single thread chromosome. Like I said, when it's one like this, it's a single thread chromosome. And then after it went through DNA replication, we have a replicated chromosome. So this is also a chromosome, but this one, to be specific, we can call it a replicated chromosome. Yes, if you say chromosome, you are still correct, but if you say replicated chromosome, then there you give us a full clarity of what you are talking about, because also this one, it's a chromosome, but this one is a single thread chromosome. So this is a replicated chromosome. And then one of this, if you see here, this single thread chromosome and then this part here are the same, but this is a chromosome. This one, when they are joined together, we call it chromatids. It's a chromatid when they are joined together. Immediately if they separate, it become a chromosome. And then when they are together like this, we call them sisters chromatids. And then they're joined by the centromere. Centromere is this red center which is hold these chromosomes together. So after DNA replication, we have a replicated chromosome or a duplicated chromosome. So these are another thing that we must know. We must be able to label this. This is a chromatids. This is a chromatids when they are together, sisters chromatids, and then sisters chromatids together with centromere, they are chromosome or replicated chromosome. And then another thing that the examiner wants us to know, we must be able to differentiate between haploid and diploid cell in terms of chromosome number. We must also differentiate sex cell, which are gametes, and somatic cell, which are body cell. We must also be able to differentiate, and then we must also be able to differentiate sex chromosome, which are gonosome and the autosome. So all this information is here. Here we have human karyotype, which is diploid cell. Diploid cells just say you have a pairs of chromosome. We have chromosome from our father and then we have chromosome from our mother. So we call it a diploid cell. And then somatic cell. Somatic cell are body cell. Body cell are diploid cell. And then after this cell goes through meiosis, we have haploid cell. So one function of a meiosis is to produce haploid cell. So if this cell goes through meiosis, then we will have haploid cell, which is N, and then diploid cell is 2N. Haploid cell has only single chromosome. The chromosome are not in pairs. If you can see here, we have one which is single, also two, they are single. So that is mean this one went through meiosis and then we ended up with one chromosome. And then chromosome number 23, we call it sex chromosome or gonosome. So this is number 23. This is 23. So these are the chromosomes that determine the gender of the organism. Like here, if it's X and X, the organism is a female. If it's X and Y, this organism is a male. So you must be able to determine to differentiate. So like here, we have haploid, which also have the single chromosome. The chromosome are not in pairs. So if it happened that this haploid cell contains X here, and then it's from a male. So when it fuses with an ovum, if this thing is for a male and then it's an X here, the zygote that is going to be produced will be a female. But if this is a male and then the sex chromosome is Y, then the zygote that will be produced is going to be a male. So a gender of an organism is determined by the cell from the male. So our father are the ones that determine our gender. So but they are not in control, but their cells are the ones that are determining our gender. So these are the things that you must be able to interpret. So this is haploid, which is a sex cell or a gamete, and then this is a sex chromosome, which is a chromosome number 23. Then another thing that the examiner wants us to know is the revision of the process of mitosis. We did mitosis in grade 10. So mitosis is divided into 4 phases. We have prophase, we have metaphase, we have anaphase, we have telophase. And then cytokinesis is not part of the mitosis, but it helps to split the cell into 2. So when the cells are split into 2 cells, this process is called cytokinesis. That's like here you see the cytoplasm is start to split. So it's the process of cytokinesis. But the phases are prophase, metaphase, anaphase, and the telophase. So you must be able to explain, what happened in prophase. In metaphase, you must also be able to explain in anaphase and in telophase. So that's another thing that the examiner wants us to know. Then now we go to the processes of meiosis. So the things that the examiner wants us to know in the processes of meiosis, we must know the definition of meiosis. We must be able to define meiosis. So meiosis is a type of cell division that results in 4 daughter cells, each with half the number of chromosomes of the parent cell. So see here in the sketch, we have parent cell. The parent cell have 4 chromosomes. Then meiosis takes place. We have 4 daughter cells, and the cells each contains 2 chromosomes. So this is the function of meiosis. Meiosis halves the number of chromosomes from the parent. So it moves from diploid to haploid. Then another thing that the examiner wants us to know, we must know the site of meiosis in plant and in animals. So here I've showed something here. These are female reproductive system and then male reproductive system, and then this one is in plant. You remember this flower. So the meiosis takes place in the ovary in a female organism, and then in a male organism meiosis takes place in the testes. And then in plant, meiosis takes place in anther, which is a male part, and then it takes place again in ovary, which is the female part. So these are the sites where meiosis is taking place, in an anther and then in an ovary, which is one is male part, one is female part. This will produce pollen, and this produce an eggs. And then here this produce sperms, and this produce egg cells. This is the site where meiosis is taking place in animals and in plant. Then another thing that the examiner wants us to know, we must know that meiosis is a continuous process, but the events are divided into different phases for convenience. Like we have event of interphase, and then we have event of meiosis 1, and then we have events of meiosis 2. So the events of interphase, during the interphase, it's where DNA replication is taking place. We must know that DNA replication takes place during the interphase. What happens is we have a single thread chromosome. Then when DNA replication takes place, we end up with double. If you can see here, these are double or a replicated chromosome. So the sister chromatids are held together by centromere. So these two are sister chromatids. They have the same DNA, they are equal in size, everything is the same with this chromatids, sisters chromatids. So they are the result of DNA replication. So it was one, now we have double. So, and it takes place during interphase. So you must know that let's see, DNA replication takes place, a chromosome which are single thread becomes double, and then each chromosome will now consist of two chromatids joined together by centromere. So it's just what I've just showed you. So, and then DNA replication helps to double the genetic material so that it can be shared by a new cell arising from cell division. So this is the purpose of the DNA replication. It doubles the genetic material so that it can be shared by the new cells arising from cell division, arising from mitosis or meiosis. And then we have the events of meiosis. We have meiosis 1, and you must be able to explain meiosis 1 using diagrams. So I have a video for that. I put a link, you can click on the link, and then the event of each phase of meiosis 2. Again, we have meiosis 2. So here is the process of meiosis. We have meiosis 1. Meiosis 1 contains prophase 1, metaphase 1, anaphase 1, and telophase 1. So you must be able to explain this process here using sketch, because during exam they will show you the sketch and ask you which phase is this. So you must be able to interpret these phases using sketches. So, and then we have meiosis 2. Meiosis 2 contains prophase 2, metaphase 2, anaphase 2, and the telophase 2. So this is what meiosis do. Meiosis halves the number of chromosomes, but this process, all of this process are taking place from here until we produce these 4 daughter cells. So they went through this process before we can have 4 daughter cells. And then another thing that the examiner wants us to know, the importance of meiosis. So the importance of meiosis is the protection of haploid gametes. I've showed you the haploid gametes. Haploid gametes, they are not in pairs. The chromosomes are not in pairs. So it's the result of meiosis is to produce haploid gamete. And then another function, the halving effect of meiosis overcome the doubling effect of fertilization. So this is the examiner wants us to know that the reason why meiosis halves the number of chromosomes is because it wants to overcome the doubling effect during fertilization, thus maintaining a constant chromosome number from one generation to the next. So let me use this to explain. Male, we have 46 chromosomes in total, and then female have 46 chromosomes in total. So if maybe a male and a female, they conceive a kid, so after fertilization, if meiosis do not take place during fertilization, they were going to be 46 chromosome and 46 chromosome, which will make 92 chromosome. So now this zygote will have 92 chromosome, and then while the parent have 46 chromosome. So this chromosome, they will have double. So because of meiosis halves the number of chromosome, so now we have 23 chromosome, and 23 chromosome, they are half, and then the zygote, we end up with 46 chromosome, which is 46 chromosome from the parent. So meiosis half the number of chromosome. So we ended up overcoming the doubling effect. So they won't double, it won't be 92 here, it will only be 46, the same as the parent. So this is another thing that you must be aware of, the reason why we go through meiosis. And then another thing meiosis do is the mechanism to introduce genetic variation through crossing over and the random arrangement at the equator. So with this one, you can watch the video of meiosis, then I will explain the process of crossing over and the random arrangement at the equator. So it helps us with genetic variation. So meiosis is the one that makes us look different, even if we are born from the same parent, but we have this different genes. And then this is the purpose of meiosis. And then we have something called abnormal meiosis. So with the abnormal meiosis, there is a non-disjunctions and its consequences. Non-disjunction is the failure of homologous chromosome or sister chromatids to separate properly during the cell division. So as we go through the cell division, there is some processes where chromosome must separate. So if they fail to separate, that process is called non-disjunction. And the consequences of non-disjunction is non-disjunction result in daughter cells with abnormal number of chromosome. So if the separation do not take place, we will end up with a cell with the abnormal number of chromosomes. So it's another thing that we must know. And then non-disjunction of chromosome at 21 position during anaphase in human to form abnormal gametes with an extra copy of chromosome. So here you must know something called non-disjunction at chromosome number 21. So I will explain it in the next slide. The fusion between an abnormal gamete, which is 24 chromosome, and then a normal gamete, which is 23 chromosome, may lead to Down syndrome. So here I'll show you something here. If this non-disjunction takes place maybe in this male at the chromosome 21, there is two chromosome, which it happened that they didn't separate. So we ended up with one extra chromosome, which are 24 chromosome, and the female it have a normal number of chromosome, which is 23. Then if they fuse, if there is a fusion between this egg cell and the sperm cell, so we will end up with 47 chromosome, which is on 21 chromosome we have 3 chromosome. And then these 3 chromosomes here, we will end up with a condition that is called a Down syndrome. So a Down syndrome. A Down syndrome is a condition that we see like on a face. This person with a Down syndrome, here there is a flat nose bridge. The bridge here is flat, and then they have small eyes, and then a small ears, and then a short neck. You don't have to know this. You just have to know that a person with a extra chromosome at 21 will end up being a Down syndrome or trisomy 21. So we call it a trisomy 21. So these are the things that you must know, that what happened when a sperm cell with extra chromosome fuses with an egg cell with a normal number of chromosome, or it's either it can happen in this female part. You find that here we have 2 chromosome and this side there is 1. We will also end up with 47 chromosome, and then the result will be Down syndrome. So it's another thing that the examiner wants us to know. And then the last thing that the examiner wants us to know is the comparison of mitosis and meiosis. So we must know the similarity of mitosis and meiosis, and then we must know the difference between mitosis and meiosis. So please watch the videos where I explain the similarities and the difference. So I've put the link so you will be able to watch them. So if you watch this video to this far, thank you very much. Please consider subscribing so that our channel can grow. Then we will make research so that we can give you a good content. Then if you are studying, I say good luck with your studies. Thank you very much.